Thank you for inviting me. It's been a beautiful day, amazing presentation. I'm going to try to be as quick as I can because I know we are running a bit late. That's an overview of my talk. I will skip probably on the introduction because it's been, small vessel disease has been introduced previously very well by other speakers. I'm going to talk a bit about the role of the neurovascular unit in small vessel disease, and then I will describe the model that we have generated in the lab using collagen 4 patients that have induced pluripotent stem cell, and discuss some of the results. So as I say, we know the cerebral small vessel disease are important disease, and you know that unfortunately, despite being important, there is currently no treatment, and the reason is because we don't fully understand the underlying mechanism, and also there is a lack of relevant model. What we know, though, in recent years, there's been a lot of study on monogenic form of small vessel disease. These are forms that are caused by single gene disorder. These are mutations in specific gene that include NOTCH3, HTR1, of course, collagen 4A1-A2 gene. And then there is another form which is called sporadic or polygenic. This is the most common form of small vessel disease. These are two distinctive forms, but we think they share underlying pathogenic mechanisms. So especially by studying this genetic form of SVD, or small vessel disease, in recent years emerged the importance of the neurovascular unit. As you probably know, neurovascular unit is a highly specialized, highly complex structure present in the microvasculature in the brain, and is made of a cellular component, which include astrocyte, brain endothelial cells, and neural cells. These are pericyte on the capillaries and vascular smooth muscle cells on the arterial. And the specialization of the neurovascular unit function is due by the brain endothelial cells monolayer, which express this complex of protein called tight junction, which literally seal the cells together, preventing passage of substance between the cells. Therefore, the passage of substance in and out the brain in and out of the blood is highly regulated, and this structure is also known as a blood-brain barrier, and this unique structure of the brain is very important to protect the brain from toxic substance and viruses, as we know. There is another important component. This is a cellular component, as we mentioned earlier on, a basement membrane made of extracellular matrix protein and related pathway. Of course, very important component is collagen 4, is one of the most abundant protein in the basement membrane, which is produced by collagen 4A1-A2 gene. And we think the ACM is very important, not just because it offer a structural support to the vessel wall, but important also in mediating the communication between the different cells that form the neurovascular unit. So what's happening in small vessel disease? Neurovascular dysfunction is seen as astrocyte detachment, loss of neural cell, loss of tight junction, and breakdown of the extracellular matrix. We don't fully understand how this cascade of event occurs, but we know the end results is an increase in blood-brain barrier leakage, which is seen on MRI in patients. And again, we have focused our work on understanding the mechanism that leads to extracellular matrix breakdown, contributing to neurovascular dysfunction, and being collagen for an important player. We have developed model for collagen 4 to study the mechanism underlying this disruption. So we think to study this disease is important to develop models which are relevant to the disease, and human model, they carry the same mutation that we see in patients with the same genetic background. So for our study, we have recruited two individuals, one with a collagen 4A2 mutation and one with a collagen 4A1 mutation. Both have cysteine type of mutation, which disrupt the structure of collagen 4, potentially alter the function. So what we do with this This patient is generate human-induced pluripotent stem cells, so iPSC. If you're not familiar with the iPSC technology, I will describe quickly with this diagram on these slides. So once we have recruited a patient with the causal COL4A1-A2 mutation, we take skin biopsy, skin cells, and then we turn these cells through a process called reprogramming, into induced pluripotent stem cells. These cells behave like embryonic stem cells. They can renew indefinitely. But also, if you give the right factor, they can turn in all the cells of the body. And we can also replace the mutation in these cells with the wild type allele using a technology which called gene editing, or CRISPR, which has been mentioned before. And so we have now the disease iPSC and the isogenic corrected line that we can use as a control. And we can differentiate, so turn this line into the cells of interest, brain endothelial cells and mural cells. We can now be in co-culture in our disease in a dish model, which is a transwell model. This is a simple setup, but in a way mimic the neurovascular unit with the brain endothelial cells, which are placed on the top of the transwell, representing the lumen of the vessel, and the mural cells at the bottom of the transwell, representing the brain side. And we can use this model for mechanistic study and also potentially to develop platform for drug screening and discovery, with, of course, the aim to identify new potential target for therapy. So this is how the model looks in real life. And in these slides, I'm going to show some of the assays that we've been performing using our transwell in vitro model. So first of all, we look at the tight junction distribution on the surface of the brain endothelial cells, and this is mainly by immunostaining. We look at, in particular, occludin and claudin 5 distribution and abundance, because we know are important, as I mentioned earlier, in regulating the function of the brain endothelial cells. We also perform functional assays. These include the transendothelial resistant, or TEER, and this is consists of a chopstick electrode method, which again inserted in the transwell and basically measure the resistance which is offered by the endothelial monolayer. And the higher is the value and the longer is maintained, is basically a representation of barrier integrity and tightness of the barrier. Another assay we perform is the site-selective permeability assay, which consists in adding fluorescent molecular specific size at the top of the transwell, and then measuring the molecule that cross the brain endothelial monolayer. And I will describe this assay a bit later in the talk. Looking at results in our iPSC-derived collagen 4 model, some are described here in these slides. We see, first of all, looking at tight junction distribution on the surface of the endothelial cells. And this is by, as I mentioned earlier, by immunostaining for occludin, here in red, and claudin 5 in green. What we see is an increase of, we see some dysfunctions and abnormality. We see an increase of tight junction discontinuity in the occludin staining, which means an increasing gap in the distribution of the tight junction. As well, for claudin 5, we see an increase in intracellular accumulation, which we don't see in the corresponding isogenic control. Also, when we look at the extracellular matrix, which is produced by the mural cells, we see a significant decrease in collagen 4 deposition extracellular matrix compared to the control. And also, we see morphological change in the shape of the cells. They express more actin stress fiber compared, again, to the isogenic control, which might indicate that the cells are stressed, they have difficulty in attachment, and that could then lead to the other phenotype that we observe in these cells, which is an increase in cell death in apoptotic event. Shown here by this graph, there is represented staining for FISH annexin 5, which is early apoptotic marker. We see for COL4A1 in red a substantial increase in apoptotic event compared to the control. So ultimately, we want to see how this defect in extracellular matrix and in tight junction distribution can affect the function of the blood-brain barrier. So we performed this assay with this site-selective permeability assay with this sodium-fluorescent molecule. They are added at the top of the transwell, and then we collect the fluorescent at the bottom of the transfer across a time course of 8 hours, and we see a significant increase in blood-brain barrier permeability in both our in COL4A1 and COL4A2 iPSC-derived blood-brain barrier model compared to the isogenic control, which is indicative of blood-brain barrier breakdown as we see in patients. Now, a limitation of this model is that there is a lack of cell-cell interaction. So we want to see how the disease mural cells can mediate blood-brain barrier disruption by paracrine effect, so through the factor they do secrete in the media. For this reason, we have treated the isogenic brain endothelial cells. This is healthy endothelial cells with disease mural cell condition media. And upon treatment, again, we see an increase in tight junction discontinuity, an increase of tight junction abnormality shown here, again, by occluding staining in red comparing to the control. And also what we see when we perform TR measurement across several days, we see consistently a reduced TR value in the treated cell compared to the untreated, suggesting the mural cells, the disease mural cells, contribute to the phenotype. As I mentioned early, a limitation of the transwell is there is no cell-cell interaction, as we know is very important for is physiological, first of all, and very important for neurovascular unit function. So we've been developing last few years a blood-brain barrier on a chip model, which consists of a microfluidic chip where we can culture iPSC-derived brain endothelial cells, mural cells, and astrocytes. In this chip, the three cells are in contact with each other, and the contact of the cells promotes the formation of macrovascular network is shown here, and these promote also expression, tight junction, a decrease of permeability. So we form something that replicates the microvasculature in the brain, and importantly, this nectar, the microvascular nectar, can be perfused with dye or even like blood cells. And by perfusing with our model with FISH dextran here, we show that by switching the isogenic, so this is the model with astrocyte, brain endothelial cell, and mural cells. This is the wild type. By switching the isogenic mural cells with the disease mural cells, we increase the permeability of the vessel, again, supporting the idea the mural cell contributing to blood-brain barrier dysfunction. The next question is what is secreted by the mural cells, which might contribute to the defect that we see in our model. To answer this question, we have performed a transcriptomic analysis of the disease COL4A1-A2 iPSC-derived mural cells and versus the control, and we identified 16% of the differentially expressed gene are extracellular matrix protein. They have a different collagen, and among those there are several matrix metalloproteases, or MMP, which have been mentioned earlier on. These are important protein, important enzyme. They are important for the function, the remodeling of the extracellular matrix, but if expressed in high level, can contribute to blood-brain barrier, extracellular matrix breakdown and blood-brain barrier dysfunction. So to test this hypothesis, we had treated ourselves with cells model with doxycycline, which is quite a blunt instrument, which is because it's a PAM MMP inhibitor. Upon treatment, however, we see an amelioration of tight junction distribution in the brain endothelial cells, on the surface of brain endothelial cell. And also we see an increase in collagen 4 level in the extracellular matrix, so the, produced by the neural cells. And importantly, when we treat our iPSC-derived blood-brain barrier model with doxycycline, we see a significant decrease of barrier permeability when we use a permeability assay, sodium fluorescence assay. So another important aspect, as a final part of my talk, another important aspect that we are exploring using our model is to set up a platform for high-throughput screening, which allow to test in a time and cost-effective manner multiple inhibitor library of small molecule repurposing drugs. I think it's a really important aspect of our research. And so to do that, we have set up our site-selective permeability assay, which I mentioned earlier, in a 96-well plate format. And this is, again, allow a very simple readout just by using a plate reader. To test if this setup is effective, we have screened a small library of MMP inhibitor, MMPI. These are a small library of 13 MMP inhibitor. And upon screening, we identify 3 MMP inhibitor that effectively reduce the blood-brain barrier permeability from a high-range permeability where the disease are located to a low-range permeability comparable to the healthy control. And these inhibitors are also able to reduce the apoptotic level that we see in neural cells, so shown in this graph, compared to the untreated, we are in red, to level that are comparable to the control shown in blue. So again, this is like proof of principle that our model can be scaled up for potential high-throughput screening of inhibitor and molecule of interest. So this is a summary. I'll just show you. So we think we generate a relevant model to study collagen 4 derived small vessel disease. In our model, we show extracellular matrix abnormality, blood-brain barrier leakage, as we see in patient, and we identify MMP as a potentially contributor to small vessel disease and a potential target for therapeutic intervention. And with this, I would like to thank you all, my collaborator and my founder body, and thank you for your attention. Taking any question.
Thank you so much also for being perfectly in time. So we have gained some minutes, so we can have the questions right now instead of collecting the 2 talks. I would like to ask for, just for the benefit of the family that may be not familiar with the process of, developing drugs for rare disease. Could you ideally show the pathway from identify a drug to the final goal of treating? Give some hints for the families.
Sure.
How long is the path?
How long is the path? Yeah, I've been asked this question. So clearly what we're doing is quite basic science. These are in vitro models, so they will then, depends on the drugs. If it's a repurposed drugs, then available. If it's a new drug, then we will have to test in animal and then test if it's safe to use in human. So it could be from 5 to 10 years, probably, type of, time.
I think it's really important to, also put him in a timeline, but also to understand how difficult it is to find the right strategies. So if there are no other questions, thank you so much.
Thank you
very much.